everyone, I am Dr. Swarupa Agnihotri. Would like to invite you in lecture series of Insights of Plant. In this lecture, we are going to study chromosomal mutations or chromosomal aberrations. First of all, we will focus how this genetic material is present in the cell. Genetic material in the form of RNA or DNA is stored in the genome. This genome is distributed in the cell in the form of chromosome. In prokaryotes, it is in linear form as well as it is in circular form. Now, when it is present in circular form, we call it as plasmids. On the other hand, in eukaryotes, linear form of chromosome we will find. This chromosome is present in some sets, haploid and diploid. Haploid means half set of a chromosome and diploid means two sets of chromosome. Structurally, this chromosome in case of a eukaryote is having one centromere at the center and two chromatid arms. These two chromatid arms are short arm is also called as P, long arm is also called as Q arm. At the end of these two chromatid arms, there is a presence of a region called as a telomeres. These two telomeres are very important because they give stability to the chromosome. And centromere is very important because at this particular point there is an attachment of spindle fibers during the process of a cell division, that is mitosis and meiosis. Now we actually see what is exactly mutation means. Now then, when variation in normal chromosome structure or number of a chromosome changes, we call it as mutation. Structure means if suppose in normal case chromosome has particular length and because of a deletion there is a change in the length of a chromosome, we call it as mutation, structural type of a mutation. At the same time, when if we have one set of a chromosome and after the mutation, if we find two sets of chromosome, we call it as numerical mutation or numerical aberration. These aberrations are further classified as spontaneous type of a mutation and induced type of a mutation. Spontaneous means sudden mutation which occurs in the body, and induced means there are some external agents which are responsible for creating mutation in the body. In that, first one is called as radiation. Alpha, beta, gamma, these are the rays which can cause the mutation in the body. Next one is base analogues. There are certain bases which can replace our natural bases in the body. Some alkylating agents, pesticides, fungicides, some genes are also responsible for creating mutation in the body. Now we will see types of mutation, structural mutation. In that, first one, deletion. Deletion means breaking and deletion of particular segment of a chromosome. Next type of a mutation is called as duplication. Now in this case, particular segment of a chromosome shows a repetition, that is called as a duplication. Now in case of an inversion type of a mutation, there is a breaking of chromatid segment and after breaking it is again reunited with the chromosome, but it will invert at 180 degree to its original position. Next type of a mutation is called as a translocation, where there is a shifting of chromosome segment to non-homologous chromosome segment. So these are four different types of structural mutation. Now coming to the deletion. There are two types of deletion. First one is called as a terminal type of a deletion. Now in this figure you can see there is a presence of a chromosome and which shows deletion at end position, that is at terminal position. The end position gets deleted and due to that there is a shortening of chromosome. So this is called as a terminal type of a deletion. Second one is called as a intercalary type of a deletion. Now in this case there is a deletion of middle portion of a chromatid arm. That portion is present between end position and centromere. So that part gets deleted and which results in shortening of chromosome. Now coming to the cytology. After deletion, there are some changes in a cell cycle. You can find these changes as cytological structures. In that, first one is compensation loop, that is formation of compensation loop. Due to the deletion, unpaired chromosome shows a formation of loop-like a structure that is called as a compensation loop. Second one is called as laggards. Now these laggards are nothing but the deleted portion of a chromosome. This portion is without centromere, that is it is acentric in nature. So it is not attached to the spindle fiber of a cell and due to that it is not involved in the cell division process. It is lagging behind, so that is why it is called as laggard. Third type of a cytological effect is formation of ring chromosome. Ring chromosome, that means when there is a deletion of some part of a chromosome, that deleted part remain as it is and remaining portion of a chromosome with the centromere gets fused and that fusion produces ring chromosome. So due to the deletion, there are also some genetical effects in body. So these effects we will see one by one. In that, first one is lethal effect. Lethal means deletion of two homologous chromosome, that is some part of a homologous chromosome shows deletion and due to that there is a lethal effect in body. Second one is called as a pseudodominance. Now when two chromosomes are present and in that when there is a deletion of dominant allele and in this case there is a sudden expression of a recessive gene, so that expression is not expected, so that is called as a pseudodominance. Second one is genetic effects on human beings. So there are various syndromes which occur due to the deletion process. Now in that first one is Cry-do-Chart syndrome. Now this syndrome occurs due to the deletion of chromosome number 5. Chromosome number 5 and in that particular short arm of a chromosome, segment number 15.2 and 15.3 shows deletion. And due to this deletion, there is a syndrome called as a Cry-do-Chart syndrome which occurs in infant. Now this image shows two kids. On the right hand side, you can find a kid with a normal brain size and normal facial expression. On the other hand, the kid which is present on left hand side shows a reduction in the brain size and abnormal facial expression. So these abnormal kids, they are not having proper feeding as well as they cry like a cat on high pitch mode and they have very reduced intellectual power. In worst cases, these kids are die during early age also. Turner syndrome occurs due to the deletion of Y chromosome. Short arm of a Y chromosome has a region called as SRY region. This region is responsible for production of testis development factor. and due to the deletion of this particular region, there is no any testis developing agent, and due to that man shows female characteristic, that is nothing but the Turner syndrome. Next is Prader-Willi syndrome. Chromosome number 15, long arm of a chromosome number 15 shows deletion. Now due to the deletion of this particular chromosome, infants has problem with the sucking. They show sucking reflex problem, and due to that they cannot feed regularly. We externally provide food, and overeating causes the obesity in these particular kids. In extreme cases, these kids also shows dying. Philadelphia is disease which also occurs due to the deletion of chromosome number 22. Long arm of a chromosome number 22 shows deletion, and due to that there is the occurrence of leukemia in the patients. Drosophila also shows deletion. X chromosome of Drosophila, in particular 3C2 to 3C11 bands, get deleted, and due to that there is a formation of notch in the wings of Drosophila. Melandrium plant also shows deletion. Melandrium has XX and XY type of a sex determination pattern. Y chromosome of these plants has some segments, in that when there is a deletion of segment number 1, we find hermaphrodite type of a flowers. When there is a deletion of segment number 2, we find neuter type of a flowers. And when there is a deletion of segment number 3, we find male sterile flower. So in this lecture, we have seen types of deletion and its genetical effect. In next lecture, we will see duplication and its genetical effect. Thank you.